Professor from Norway, Inari Kursula, Professor, University of Bergen, which is going to speak on using photons and electrons to understand malaria, parasite, motility. So very real story behind photons and electrons. So Inari, the floor is yours. We are absolutely happy to have you here.
Thank you very much. I hope you can hear me.
Yes, we can.
And I hope you can see my presentation.
Mm-hmm.
Yes, I have labs in Norway and Finland, so also two smallish countries who don't have any large facilities for photon or neutron science, but yet we are quite big users of European facilities. So today I'm going to tell about how my lab is using photon science and also not neutrons but electrons to understand the malaria parasite motility. Motility, so the ability to be able to move from one place to another, is very important for all of us, from us humans to other living creatures, all the way to small single-celled organisms. And we humans use muscles for this, but of course things like malaria parasites, which are actually just composed of one single cell, they don't have muscles, but they use other kinds of mechanisms for this, and yet these mechanisms are based on very similar biochemical functions as our muscle contraction. And a major protein important for all kinds of motility is actin, which is depicted here in cyan color. And my research is focused around actin-based motility, and this is what I'm going to talk about today. Here you see actin in its monomeric form, so a single molecule of actin, but actually this is not the functional form of the protein. Its functionality is based on the fact that it can form these beautiful long filamentous structures which are very regular. And here you can see an electron micrograph depicting these filaments, which in our muscles can be tens of micrometers long. Excuse me. But in the malaria parasites, they are very, very short. I'm sorry for the dog, we just have to ignore him. Actin and its ability to produce force for motility has been studied extensively in many organisms, and typical model organisms come from a very narrow branch in the evolutionary tree, and that includes animal actins and yeast actins and some other organisms. But very little actually is known about the function of actin in other more simple organisms. So that's why when I started my own research a bit more than a decade ago, I decided to start working on something which is very different and which maybe not so many people are working. And I decided to choose the malaria parasites as my model organism. The malaria parasites, as I said, they are just a single cell, but they have a very complicated life cycle. They need two very different hosts to complete their life cycle. They need a human or another vertebrate, and they need a mosquito. Of course, because the life cycle is a cycle, you can start it from anywhere, but I tend to start explaining the life cycle from here when the infected mosquito takes a blood meal from a host. That's how it delivers the parasites into the body of the host, in this case a human. And the form that the parasite delivers the mosquito delivers to the human is called a sporozoite. And these sporozoites require a certain form of motility, enabled to first get from the skin of the host into the blood circulation, from the blood circulation eventually into the liver, where they go through their first so-called exoerythrocytic cycle of reproduction, and then again to the blood circulation and start invading red blood cells. And this is already a different form of the parasite. During the blood cycle, the erythrocytic cycle, the parasites form gametocytes, which then are taken up by a new mosquito. And in the mosquito, then the parasites complete their reproduction with a fusion of the gametes, of the zygotes, and formation of first these things called oocytes, which again have to move to get out of the gut into the salivary glands of the mosquito. And then they can start a new cycle infecting a new human host. And during this life cycle, as you can see, the parasite looks very different at different stages, and it requires to move in very different environments between two very different hosts. So my research is aimed at understanding how this really works, how the parasite reaches the goal of moving inside the host organisms and invading their host cells. Here you can see sporozoites. So this is the first form of the parasite that the human host encounters. And here they are gliding on a glass slide. And their motility is indeed called gliding motility because, as you see, they glide very, very smoothly and very rapidly without any visible protrusions from the cell, without any kind of muscle force. It's very hard intuitively to understand how the force for this kind of motility is produced. These are also among the fastest moving eukaryotic cells. They move from 1 to 3 micrometers per second. So how is force for gliding produced? Actin is known to be involved, but it's very different from other types of cell motility. Here you see another type of single cell moving. And in this case, actin is producing the force for motility. And it's somehow intuitively fairly easy to understand that the ability of actin to form these long filamentous structures enables it to grow into these filaments at the edges of the cell and push the cell forward. But this is completely different in the parasite. You see that there is nothing like that happening here. There is no cell edge which is protruding from the parasite. So how does this work? For about 15 or close to 20 years, there has been a kind of a working model of how parasite motility could work. And this involves actin filaments, which you can see here as yellow, acting as kind of tracks for a motor protein called myosin, which moves along actin and can produce pushing forces. Also, the whole motor involves many other proteins. It's linked to 2 biological membranes, the plasma membrane, so the outer membrane of the parasite, and something called the inner membrane complex of the parasite via many different proteins. And via the plasma membrane, it is in contact with host cell surface receptors of, for example, the human host cell. And the hypothesis that we have had for a bit more than a decade is that myosin, indeed, it performs this so-called power stroke. It steps along the actin filament, pushes the actin filament backwards towards the rear end of the parasite, and that results in a net forward movement of the parasite. Of course, this mechanism is in reality much more complicated. For example, the parasite also needs to release, so there have to be proteases in the membrane which cleave the entire machinery off from the membrane. But this is the basic hypothesis which we have. Actin filaments serve as tracks for myosin, which uses its power stroke stepping movement to produce the force for this gliding motility. It has to do this in a very fast and efficient way to reach this, what you see here, the circling of the sporozoites. So the aims of our work in my lab are to understand the mechanism of actin polymerization, how actin forms these filaments, the tracks, and how actin and its regulatory components have evolved in the malaria parasite and also in eukaryotes in general, because they are so far apart from us, for example, in the evolutionary tree, like I showed. We also want to draw a complete molecular picture of the entire glidiosome, so the whole protein complex which is responsible for this movement, and to understand how force is generated by the myosin and the whole machinery and transmitted into this smooth gliding that you saw in the previous slide. And of course, because we're dealing with something like the malaria parasites, which are such deadly parasites affecting large parts of the globe, we want to also evaluate whether the glidiosome, motor, and the actin and its regulatory components could serve as candidates for drug and vaccine development against malaria. So what is so special about Plasmodium actins? Why to choose this system? Actins are in general very conserved proteins, so they are nearly identical between all eukaryotic cells. But the Plasmodium actins have a rather low sequence identity to us, humans, our actins, and also between the 2 actin isoforms which are present in the malaria parasite. And when I say low, this is low in the actin field, so it still is close to 80% identity. But for example, yeast and human actins are way more than 90% identical with each other. A very peculiar feature of the parasite actins is that they form very short filaments, which are in the range of about 100 nanometers, whereas the filaments that I showed in the beginning of this presentation, they can be, as I said, tens of micrometers long. We also found out already a couple of years ago that ATP hydrolysis and phosphate release pathway seem to be different in the parasite, and I'll go more in detail into this in a bit. Also, we have shown that polymerization is not governed by ionic strength in the same way as in canonical actins, and that stable dimers are a significant species in addition to these filaments, which has not been seen for any other actin. But today I'll mostly concentrate on the question, why are the filaments so short, and how does ATP hydrolysis and phosphate release work in these parasite actins? Before I go into the parasite actins, I have to explain something very fundamental about actin polymerization in general, because not all of you might be familiar with it. So here you can see a very schematic view of a canonical actin molecule. You can maybe recognize it from the picture I showed before, and a molecule called ATP is very important for the function of actin. And in this film, ATP-loaded actin is colored in yellow. Actin is actually an enzyme, and it catalyzes the cleavage of the bond between the two terminal phosphate groups, so the catalysis of ATP hydrolysis into ADP. But as a monomer, it's a very poor catalyst, so it needs to go to the filamentous form to perform this catalytic function. And when actin concentration gets high enough, there's a spontaneous formation of a nucleus, which initiates polymerization. And when actin polymerizes, it goes through a conformational change, which activates the hydrolysis of ATP to ADP. And ADP actin here is depicted in blue. And when ATP is hydrolyzed, then that induces another conformational change, and actin filaments are destabilized and depolymerize. So this is how it functions in canonical actins, and that's the basis for this directional growth of actin filaments and how the filaments can, for example, push cell edges forward. But this seems to, as I said, work a bit differently in parasite actins. We discovered already quite a few years ago that in Plasmodium actin, they actually form these short oligomers in addition to the filaments, and they form them very, very quickly, and that this is actually linked to the hydrolysis of ATP. And we also found out that ATP hydrolysis does not require the filamentous form to happen. So it happens also maybe already in monomeric form, but definitely then in the short oligomeric forms. And this is really interesting because that would kind of be against the dogma of how this directional growth of filaments can work. We then solved some crystal structures using synchrotron radiation of Plasmodium actin, both with ATP and with ADP, and we found out that there are some differences in the active site. For example, there is this asparagine residue, which is unique to the parasite actins. It's hydrophobic, so a very different residue in, for example, human actins and basically all other actins. And this asparagine residue actually moves when you go from the green ATP form to the yellow ADP form in this case. There are also many other things that move in the active site, and this we can see in the next slide. Let's see here. Here's an overall view of movements that happen when hydrolysis from ATP to ADP happens. And we paid attention mainly to this loop, which you can see on the foreground here in yellow, because this loop makes a large movement upon hydrolysis from ATP to ADP. And in the lower film, you can see some of the more detailed movement of side chain. For example, here, the movement of this asparagine residue that I showed in the previous slide. We figured out that if we block this movement of the loop, maybe then we can affect ATP hydrolysis. And we thought that if we can actually block the movement, maybe we can stop ATP hydrolysis or at least slow it down. So we made a mutation introducing a large residue instead of a small residue close to this loop. And that would be an alanine to tryptophan mutation, which we thought that that would block the movement of this loop. We also mutated another residue, namely this histidine on the other side of the loop, thinking that might also affect the movement of the loop on the other side. So we measured phosphate release rates from these mutants, and actually it worked completely opposite to our hypothesis. So this mutant, which we thought would slow down or maybe even block phosphate release, this was actually a so-called hyperactive mutant. And the other mutant, which we thought might actually work faster, that actually was a completely or nearly completely dead mutant. So we had to look into this a bit more in detail. And we called this loop Aloop, the one that is moving a lot, because it has this aspartate and arginine residue, and there was no loop called the Aloop yet in actin. There are many loops named with different letters. And here you can see the residues that we mutated. There's this small alanine here, which we mutated to a tryptophan, a large residue, and there's this histidine over here, which we mutated into a glutamine. In our original crystal structure, which was in complex with calcium, which is a non-physiological state of actin but helps keep it monomeric, this was the position of the A-loop. So the A-loop is interacting with this histidine residue. But we decided to determine more physiological crystal structures, meaning we determined structures in the presence of magnesium instead of calcium, because in the cell actin is in complex with magnesium, and this would be the active state of the protein. And in complex with magnesium, we got a mixture of ATP and ADP because the hydrolysis starts to happen in the presence of magnesium. And there we actually see 3 different conformations of this loop. And we call these conformations 1A and 1B, which are closer to this histidine, and 2A, which is this loop interacting with this lysine residue over here on the other side. We also eventually got a pure ADP structure, and here we see that the loop is even more interacting with this lysine over here, and the interactions with this histidine are even weaker, and the conformation 1A is missing completely. What about the mutants? How do they then look like? We saw that on the structural level, our tryptophan mutant, it worked exactly like we presumed it to work. So it locked this loop into this position. So we only see the conformation 1B. And the other mutant, this glutamine mutant, that is kind of interacting with both, but it still is only one single conformation. So you remember that this conformation is hyperactive and this conformation is almost completely dead. If you look at actually what happens in filament when we have afterwards also determined high resolution cryo-EM structures of the filament, you actually see that this loop is closer to this lysine over here, but there is another residue that comes into play in the filament conformation as well. We also looked into what the A-loop mutants do for the filament length. And whereas the wild type actin, the parasite actin, mainly forms these very, very short filaments and only a couple of very long filaments, we saw that, like we would have expected, this mutant, which is hyperactive, it forms long filaments, they are more stable, and this mutant, which is catalytically almost dead, that doesn't form filaments at all, or some very short filaments. So we can actually say that the movement of this A-loop is linked to phosphate release and filament stability in the parasite actin. This movement of the A-loop, it has not been seen in any of the canonical actins, but it may still happen, but it's probably not as favorable due to two small amino acid differences. This lysine 270, which is the other interaction partner of the A-loop in the parasite actin, is a methionine in canonical actins, which would reduce the favorability of the loop interacting with this residue. And on the other side, the histidine residue is methylated in muscle actins, which has a large impact on the properties of this histidine residue. So we can conclude that it's these small differences that increase the mobility of the A-loop, and the mobility increases the phosphate release and decreases filament stability. So that part of my talk dealt with how we used photons, photon science to learn about actin. But what can we use electrons for? We cannot crystallize the real filamentous form of actin because they form these long filaments which are not regular in length, and they cannot be crystallized. They are way too flexible for that. So we need to use a technique called cryo-electron microscopy for this. And we have very recently determined a cryo-EM structure to a very, very high resolution of the parasite actin filaments. As you can see from this map here, regions of the actin are close to 2 angstrom resolution, and even the lowest resolution regions are about 2.5 angstrom resolution. So we're getting close to, very close to atomic resolution already with this technique. So what can we learn from this? How far can we push the resolution? We can see water molecules in the core of the actin, and this is a very interesting string of water molecules because we actually think that this could be an alternative path for the leaving phosphate group, because the path leads from the phosphate, which is supposed to be hydrolyzed, towards the solvent, as you can see here, and this is the cavity shown in red. When the actin is in its monomeric state, you can see that this cavity is short. It does not extend all the way to solvent, so it actually needs the filamentous form for the cavity to open all the way to the solvent. And I see that I'm running out of time almost, so I'll start with my summary now. So Plasmodium actin, it follows the classical nucleation elongation pathway for its polymerization, for its ability to form these long filaments, but it has this mechanism by this A-loop, which leads into destabilization of the filaments and a very high fragmentation rate, which leads to the short filament length. There are the subtle amino acid changes, which I said, the lack of methylation of this histidine, and the methionine residue replaced by a lysine in the parasite, which enable the fast switch between the A-loop, between stable and unstable filament conditions. And a similar mechanism is possible in canonical actins, but probably this movement happens at a lower frequency. And we also discovered by cryo-EM that there is a channel in Plasmodium actin, which is gated by 2 residues, which could be the path for the leaving phosphate. So what are our future perspectives? So next, I mentioned already briefly in the beginning, there is a second isoform of actin in the parasite as well. So we want to study that in more detail as well. And we already have high resolution cryo-EM structures of actin 2 as well, which we are currently analyzing. We also want to perform mutagenesis studies, both in the active site and along this possible leaving route for the phosphate. We want to determine structures with different nucleotide states, and this is what we are already doing, and hopefully also trap the leaving phosphate, which is probably very challenging because once it is hydrolyzed, it probably leaves very quickly. We want to achieve even higher resolution, both using crystallography and cryo-EM, and we also want to look in more detail into the catalytic mechanism. So all this work would not have been possible without my research group, and I want to thank every member of my research group. A lot of the work that I presented here was performed by Esa-Pekka and Juha, who have already left my group, and the actin project is now actually mainly continued by Eze here, who is a new postdoc in my group. And because this meeting is dealing with these large infrastructures, I of course have to once again underline that none of this work would have been possible without synchrotrons, without access to these infrastructures. And we are mainly using DESI and the EMBL beamlines over there, diamond light source, and we have also access to cryo-EM via Instruct program. And in the end, I'd like to thank you and take any questions that you might have.
Thank you. Thank you very much, Inari. We have many thanks for interesting story, interesting research, and a couple of questions either. One of them is, what would be your advice to new scientists that only start using photons and electrons for research from administrative and organizational perspective? That would be the question.
Yeah, I think it's actually fairly easy to get access to infrastructures. The synchrotrons have open calls. Instruct has open calls for cryo-EM research. So I would recommend you just to look at the websites, look at the calls for proposals, and if there are things which are unclear, it's always good to contact the beamline scientists and ask them to maybe give you tips for preparing your call. But I think you should just take the initiative and apply for beam time or apply for microscopy time.
Thank you. The other question is maybe the same, but a bit more negatively formed. What was the most difficult at the beginning of the research, where the most of your own energy was going to? Like, what was the burning energy going where at the beginnings of research?
In the beginning, in the kind of research that we do, biological research, typically the big threshold in the beginning is to get the samples and get the samples optimized. So we spent actually several years before we could make enough of the parasite actin for all these experiments. So I think we spent probably 2-3 years just trying to get the samples for the measurements. And that's definitely the biggest hurdle, I would say. So when you can make your samples, your biological samples, then it's just to go and measure, and results might be surprising, might be difficult to interpret, but it's fairly easy to get to do the measurements.